Hi there, welcome to lesson 7.15. This lesson is about predatory and omnivorous nematodes, a group of nematodes that consumes other nematodes. And while predatory nematodes are extremely essential in soil ecosystems, majority of the studies that have been done on them and which will be used in this lesson are focused on their role as predators of plant parasitic nematodes. So we will look at their potential as biocontrol agents in the agricultural context. Okay, so let's jump into lesson 7.15. Okay, so predatory nematodes are divided into four major taxonomic groups or orders, and that includes Mononchida, Dorylaimida, Diplogasterids, and Aphelenchids. And they all have a distinct feeding apparatus, distinct feeding features in their mouthparts, feeding mechanisms, as well as food preferences. And I must say that the only reason why predatory nematodes are studied in the first place is for their potential as biocontrol agents for plant parasitic nematodes in agriculture. It is a shame because these organisms are extremely important in soil ecosystems and they only exist at higher successional stages of soil biodiversity and therefore soil health. So these organisms are one of the key components to study in order to determine soil health. But they are being studied in agriculture because of their potential to control plant parasitic nematodes. Here we have a beautiful mouth with pearly whites of Predatory pacificus. So the various predatory nematodes have specific feeding mechanisms that they use in order to feed on their prey. And they feed in a particular sequence, and that includes encounter with the prey, first of all. That can happen through the recognition of prey via some molecules that the prey releases called pheromones, as opposed to a chance encounter. Once the encounter occurs, there is an attack response in which the nematode uses its head to probe its prey and rubs its lips against the prey. There are also movements of the feeding apparatus and esophageal pulsations that accompany this attack response. And this is in order for the nematode to position itself at a right angle when it's making contact with the prey to allow for the attack to occur. And this is where the nematode cuts or penetrates its prey's cuticle or the skin via a stylet, a needle-like apparatus that pierces the cuticle, or they can have one giant tooth or one big tooth, which they use to cut the prey open, or have more than one tooth, so they have some teeth that vary in size and location within the mouth. And this allows for biting the prey or for shredding the prey. Some of these nematodes use extracorporeal digestion. This is where its prey is digested externally through the excretion of esophageal gland secretions. So this is like saliva type of liquid that contains enzymes and digests the nematode's insides. And sometimes there is a toxin involved as well to paralyze the prey. And when extracorporeal digestion occurs, the contents of the prey is then sucked up, usually via a stylet. And then lastly, ingestion occurs. And this might mean that the whole nematode is engulfed in the case of a small nematode or a juvenile nematode. Or if it's been shredded, these shreds can be ingested. Or if it's cut into pieces, pieces will be ingested. Or otherwise, the contents is sucked up and the cuticle is left behind. And this just depends on what kind of mouth parts the predatory nematodes have. The image on the right shows Predatory pacificus feeding on C. elegans, and this is a screenshot from a video you can find on YouTube by the Max Planck Institute for Biology. So let's first of all look at the Mononchids. They have a strongly sclerotized mouth, so that means that it's quite rigid, so it's able to deliver a hearty bite into its prey. They have this one tooth here, or they also may have large puncturing teeth or many small grasping teeth. So it just depends on the species of the nematode as to the mouth parts that it has. Several species feed extensively, but not exclusively, on plant parasitic and other nematodes, and they're able to cut the nematode cuticle and suck their contents. Some have an engulfing feeding apparatus, so some can actually swallow smaller prey or cut large prey into pieces and swallow the pieces. Their life cycle is between 15 to 45 days, and this varies with species, and they have efficient predatory capabilities, but their use as a biocontrol agent is limited because they are quite intolerant to environmental fluctuations, have low reproduction rates, and a long life cycle. They are also cannibalistic, so they can eat other mononchids. And here we have the genus Mononchus in this image. The Dorylaimids are omnivorous predators, and they are the most ubiquitous of all the predatory nematodes in all soils, across all climates and habitats. 3 or more genera can actually be present in one place, even in one field, and there are 200 to 500 million of these organisms per acre in some soils. They pierce their prey via a hollow stylet called odontostyle, and they suck up the contents of the prey. They also have a mural tooth as well, which pierces or splits the prey. So it's interesting because some of these nematodes actually have teeth and stylets at the same time. They suck up the digested prey via the oral aperture, so the mouth opening, and they have a life cycle of between 3 to 6 months. An exception is Labronema vulvapapillatum, which has a life cycle of 36 days. A species of Eudorylaimus feeds on eggs inside the cyst of Heterodera, so some of them actually are egg feeders. And as a biocontrol agent, they have efficient predation rate and wide predation range, so they can consume several species of plant parasitic nematodes. They display prey preference as well, and also engage prey searching, attraction, and several of these nematodes will aggregate at a feeding site. So these are all beneficial attributes of what a biocontrol agent should possess. Here we have an image of a Labronema feeding at the posterior region of Panagrellis. Diplogasterids are also omnivorous predators, and they are abundant in decomposing manure. They have a smaller buccal cavity, and this just means the mouth, with various teeth sizes and positions. They feed on nematodes, bacteria, and other soil microbes, and have a life cycle of between 8 to 15 days when feeding on bacteria. A species of Diplenteron and Butlerius switch to bacteria if prey nematodes are absent. They are more prey selective than other groups of predatory nematodes, and for example, Odontopharynx longicudata has been noted to consume 6 out of 17 prey species present, but it had 100% preference for Anguina pacifici. So some of these nematodes will just hunt their preferred prey, but might also consume other species of nematodes. So this suggests that there is a potential targeted prey-predator relationship that can be achieved using this nematode as a biocontrol agent. And a few species of Mononchids strongly prefer endoparasitic juveniles versus ectoparasitic juveniles of plant parasitic nematodes. Mononchids longicaudatus has been shown to reduce root knot nematodes and increase plant growth and root biomass when used as a biocontrol agent. And These guys in general possess chemotaxis, so the ability to detect chemicals in other nematodes and therefore target their prey. They have high predation rate and are easy to culture on bacteria. They display prey specificity and also have a survival stage called the Dawa larva, where they can actually go into a suspended stage that allows them to survive in unfavorable conditions. And in the image on the right, we have an example of the genus Buclarius. Another member of the Diplogasterids, called Pristionchus pacificus, displays a phenomenon called phenotypic plasticity, and this means that it can have mouthparts that are adapted to feeding on bacteria or specifically to feed on nematodes, and this is a response to changing in environmental conditions. So microbial feeding mouthparts are termed stenostomatus. This is what it looks like here on the right. But when the culture in which they are growing is aged or prey is present, these nematodes will develop predatory or eurystomatous mouthparts in order to prey on the nematodes that are present. So these guys are a model system for the phenomenon we called feeding dimorphism, which is this ability to consume very different food sources based on two distinct morphological features of their mouthparts. And the reason why they are a model system is because they have a short life cycle and are easy to manipulate experimentally, and also they are quite conspicuous. So here we have Pristionchus pacificus feeding on C. elegans in a video you can find on YouTube, as mentioned earlier. Aphelenchids are also omnivorous predators, and they have piercing and sucking mouthparts. The genus Cyanura has a needle-like stylet that penetrates its prey and then uses digestive enzymes that have toxins in them that cause instant paralysis. And here is the stylet of the species Cyanura shigaensis and its other identifying features. They can feed on small to large prey and have a life cycle of between 3 to 6 days. So they have a high reproductive potential and are easily and rapidly cultured on fungivorous nematodes. And they are considered as most suitable biocontrol agents. Another aphelenchid is the mycophagous predator Bursaphelenchus sinensis, in which some adult females develop predatory stylet, lip and pharyngeal glands that contain digestive enzymes, and they pierce the Mycophagous acrobaloides species, ingesting its contents. And here is an image portraying that. Interestingly, they are not induced by potential prey presence, but when there is a reduction in their fungal food source and an instigation of a starvation signal, this dimorphism occurs in the next generation, where the females develop the ability to consume nematodes. And it has been found that the predatory to mycophagous feeding style ratio is greater in older cultures, in media where there is a reduction in nutrients. So this is media and fungal species dependent as to what happens in terms of the dimorphism being displayed in this species. However, when predators are fed on fungi, so when they are omnivorous, they produce mycophagous offspring. So obviously the species reverts back to this mycophagous lifestyle once fungi are present. And this is a display of ecological adaptability as a result of environmental changes in the soil ecosystem. So there is a lot of research as to the potential of predatory nematodes as biocontrol agents in agricultural systems. And there are several prerequisites that need to be fulfilled in order for the nematode to be considered as a good biocontrol agent. And this includes prey searching and specificity for target organisms. So, for example, the Mononchids generally lack prey specificity and are also cannibalistic. So it does reduce their biocontrol agent capabilities. Diplogasterids and Aphelenchids display some specificity to their prey, And it is essential that there is predation efficiency as well as broad spectrum efficiency, meaning that if the nematode can actually consume more than one species of plant parasitic nematodes, it is deemed as an efficient biocontrol agent. Also, some, as we have learned, display possessed toxicity in their saliva or in their digestive enzymes, and so this is a bonus because this ensures that predation is successful. Other aspects of suitability include life cycle duration and longevity. So if the nematodes can live for longer, they can act as a biocontrol agent for longer, and if they have short life cycles, it means that there is high level of reproduction and also that they can be cultured easily to produce enough to actually be able to disseminate them as biocontrol agents. Another important factor is high reproductive potential, and this is displayed by Diplogasterids, and this group also has a short life cycle, which makes it a good biocontrol agent. It is also very important that they can survive and adapt to farm practices and ecological conditions within the soil ecosystem. So if there's a lot of plowing going on, for example, these larger nematodes actually will not survive the ordeal. So in order to be used economically, there's got to be a transition in farm practices that are more conducive to the survival of these organisms. Another important factor is that the nematodes can be dispersed across the soil ecosystem and that they will persist in that ecosystem. So this is where dual feeding habits are important, because if the prey no longer exists due to its life cycle constraints, for example, they can feed on other organisms such as bacteria. And also they must be safe to non-target organisms. And this is where it is really important to remember that these guys will not necessarily just feed on plant parasitic nematodes. A lot of them will also feed on fungivorous nematodes, and this might actually impact or affect the nutrient cycling within the soil. And it also means that they might develop preference for these non-target organisms and therefore consume the organisms who actually want to stay in the soil. Another really important aspect is that when we introduce species that have been born and bred in the lab and they normally would not exist in that ecosystem, we are actually creating problems within that particular region. So they might come from a lab somewhere completely different to where your land is, and therefore addition of these specific species, since they've been selected to be biocontrol agents, means that they might proliferate more abundantly than the actual endemic species of the nematodes that already exist or should exist in the soil but are not there because of the practices that the farm is engaging. So as always, biocontrol is quite tricky and it often backfires, and we have many examples of this across the world. And really the consequences of using these organisms as biocontrol agents haven't been studied. So we really don't know what we're releasing and how that's going to affect the endemic or indigenous ecosystem. So this is something to be mindful of, and I will always recommend to have all the organisms of the soil food web present by taking care of the soil ecosystem, by feeding it through organic matter and the discontinuation of detrimental practices such as use of herbicides, pesticides, and ploughing, as not to disturb the ecosystem that can actually be built by just creating better, healthier soil ecosystems that will naturally develop the biocontrol properties and naturally create suppressive soils if our desire is to prevent an overexplosion of plant parasitic nematodes. But in general, these organisms are so important in soil ecosystems. They have a very unique role as a very specialized predator, and we need to encourage these higher successional nematode groups to actually proliferate in the soil if we actually want to have a healthy soil ecosystem. And here is another image of a mononchid. So we have come to the end of lesson 7.15, and our last lesson on nematodes, lesson 7.16, will focus on soil nematode community and diversity in relation to soil health and plant community. So you'll be hearing from me again in lesson 7.16. Bye for now.